Ryan, how are you today?
Doing
Thanks
all right.
for coming back.
Thanks for having me again.
So, what were your biggest findings in your research?
So the biggest thing that my research covered was probably the population genetics of this pestiferous species.
Yeah.
So to accomplish this, we went to various orchards throughout the northern New South Wales region, the Northern Rivers, and we sampled from 7 different localities, 7 different orchards. And these localities varied from 300 meters apart to over 20 kilometers apart, to really represent that distance.
Yeah.
And from each locality, we took about 30 individuals. We brought those back to the lab and extracted their DNA. We then sent this on to a company called Diversity Arrays Technology, based in Canberra, to get that sequenced. And the sequencing is a genomic technique, so looking over the entire DNA strand. And we're targeting regions of the genome that are actually fairly conserved, except for one single nucleotide, or one single base pair, which is called a single nucleotide polymorphism. So these short little regions that are conserved in the genome are compared against another genome to see if you can find those variable regions. And depending on whether or not an individual has that variable region, you can kind of see how related they are to one another.
Yeah.
So, you know, if this one has it and this one doesn't, maybe they're not related, and, you know, if they both have that same nucleotide, then they would be related.
And how many strands did you collect?
So our final result was over 22,000 single nucleotide polymorphisms,
Yeah.
Which is quite a bit of data. But that was not what was in the final analysis. We actually did have to pare that down a little bit because some of the data was degraded, and you know, some individuals had missing data. So we got rid of a lot of that, and it actually shrunk it down to about 3,000.
So how did you process the data?
It's a bioinformatics pipeline. So using computer programs like R to actually read in these large genomic data sets, and then use various filters that are programmed in to cut through all the bad data.
And so you provided us with a graph called isolation by distance. Would you care to go through that with us?
So this whole process, what we were looking at was actually isolation by distance. So it's how genetically related these populations are to one another over geographic distance. And what this normally looks like is what you're seeing on the screen right now. You should see increasing genetic differences over geographic distance. However, what we found is this, which is actually no isolation by distance over geographic distance. So that's not an expected result. It is basically meaning that individuals that were taken from 300 meters apart are as related to each other as those that were taken from 20 kilometers apart.
And you would expect the more distance between the species, the more variation.
That you would see, yes. The more divergent those populations would be in those localities. So, yeah, this basically means that these lace bugs are moving throughout the region. They're very similarly related. And there's a couple things that you know, can also explain this trend, like outbreeding. So when you have a major population explosion, often genetic diversity decreases. You know, and that's a trend that you can find in some major crop pests. So that would, you know, be something that we would expect here. But it didn't quite explain all of our results. We also found that some of these genomes were actually really similar to one another, more so than you would expect by chance. You know, almost identical sometimes.
So almost like clones of each other.
Yeah, basically we found that there could be clonality within some of these populations.
And that would suggest asexual reproduction?
That's right, or some form of asexual reproduction.
Yeah.
So that's actually a common adaptive technique for insect pests in agricultural systems. And it makes sense. If you don't have to wait for a mate, you can actually take advantage of any ideal conditions within an ecosystem or within your system that you live in almost immediately, and you can breed right away. So if there's a good boom of, your food resource, you can take advantage of that and produce an offspring without having to wait for a mate.
Yeah.
So this would pose a huge problem for your macadamia growers.
That's
Yeah.
absolutely right, yes.
So with your findings, I guess there are a lot of implications for these macadamia farmers in Australia.
Yep, there's going to be some unique challenges for them to tackle in order to control these.
So in our next episode, we'll be going through the implications for macadamia farmers, and Dr. Ryan Shofner will be joining us as well. So thank you for coming in again.
Thank you.
Yeah.
Thanks for having me.
Yeah.